Hi, biology class. This is just a mini lesson on how cells maintain homeostasis. We've been learning about that, how homeostasis is essentially when living things and then the cells within those living things have the ability to maintain a relatively stable condition. And that could be in the form of temperature. It could be in the form of like pH, which is, the acidity or alkalinity of substances inside of and outside of the organism or the cells. And also we talked a little bit about glucose levels, sugar levels, insulin levels. So there's all these like molecules too that are examples of how living things can maintain homeostasis. So today, what we're going to learn about is like how living things do that, despite the external conditions. Okay, so I'm going to teach using this interactive board, and whatever I underline and talk about, you guys need to write down in your biology notes. Okay, all right. So again, what we're talking about today is the role of buffers. So go ahead and write that down in your notes, the role of buffers. I'll try to move this camera so I can kind of talk to you while I'm doing this. And we're talking about buffers in living things, right? So fish, bacteria, plants, animals, fish are animals. And so we have this ability to still kind of decrease the impact of changes in our external environment because of natural things that we have within us to help kind of offset those changes, okay? And so those things that I'm talking about in particular for this lesson are buffers. So buffers is like this new buzzword, okay? So what we know is that our cells are exposed to many different chemical environments, possibly acidic environments or alkaline or basic environments. It could be from your food, diet, and the impact of that in digestion, which then affects your pH in your body. It could be maybe a malfunctioning of an organ, a malfunction of an organ that doesn't allow it to kind of keep levels at bay. And so we have different things then that can help offset that, and that's called buffers. So what we need to know out of this slide is that cells, and really like all living things, are working constantly. There's always like molecules going back and forth over the phospholipid bilayer, over that plasma membrane. Molecules are coming in and molecules are going out. It's working constantly, and it's working constantly to maintain homeostasis, right? In this case, we're talking about an acid-base balance, okay? So it's always trying to maintain homeostasis. It could maintain homeostasis for water balance, but for this particular example today that we're talking about with acids and bases, it is constantly trying to maintain that balance of acids and bases to make it neutral. Because quite honestly, almost all living things prefer to exist at a pH at around 7, okay? The pH scale is from 1 to 14, and in the middle is 7, right? And so that's like that neutral level. And so life generally kind of like teeters right around 7 regarding homeostasis and the ability of cells and organs to function, okay? Like specifically human blood, when you go to the doctor or whatever and they take your blood, they may take the pH of your blood. And the pH of blood should be between 7.35, I'll write it on here, 7.35 to 7.45 pH, and that's for human blood, okay? So if it falls below that or above that, then there's something a little awry, something's going wrong, where maybe the body is not providing enough buffers to actually reduce the impact of the acid or base on the pH of the blood. So if there's anything off, there are natural buffers. So if anything goes wrong, despite the external conditions, we are lucky enough to have natural buffers. And natural buffers aren't just in humans, they're also in all other living things. So when I say living things, I'm talking about trout, dogs, carrots, green beans, milk, because milk came from something that was alive. And so those things are passed on in the chemicals in it. Egg whites. So because like green beans and carrots were once a living thing, then they have cells. They're made up of cells from that living thing, which then carries with it the natural buffers that it has in it. So what it can do then is offset or reduce the impact of that change. Okay, so it can reduce the impact of that pH change. And so when we do our lab, or if you've already done the lab, what you're going to notice is that there's things that can resist the change, and there's things that you're going to test that can't resist the change. Okay, the things that can resist the change in the pH are living things, or maybe they were once living things. And we consider living things biotic. Okay, so I'm going to change the color here. We'll go to blue. So this is called biotic. Okay, again, whatever I've underlined here and whatever I've written, you need to do the same on your notes. Okay, now the things that we're going to test that didn't resist the change, like they actually changed because of us changing the pH, or they were basically non-living things, those are called abiotic. So over here, we're going to put the ones that couldn't or can't resist. That means they're non-living. Okay, so the substances that we used in the lab or are going to use, depending on which, if you're blue or orange group, the things that are not living, we're going to see that it reacts differently to the addition of acids and bases versus things that are living. And things that are not living are what we call abiotic. Okay? All right. So what we now know is that buffers, these natural buffers, and it's basically like a chemical, it actually can maintain homeostasis. It can maintain a somewhat stable condition. It can withstand or reduce the impact of change. In this case, we're talking about pH. And so by doing this, it maintains homeostasis, a relatively stable internal condition. Okay, now we're going to do something kind of fun. We're actually going to draw a molecule that our cells and actually the chemicals within our cells chemically react and form this natural buffer that we actually have inside of our cells. And it's called a bicarbonate buffer system. So this is like a little snippet of chemistry, which you guys will have next year. So our buffer, the human buffer, sorry, messed up that H, our human buffer comes from what we call a bicarbonate buffer system. Okay, and it's a really cool molecule. We're going to go ahead and draw it for fun. So it has an oxygen molecule. It's double bonded, okay, with carbon. So since there's carbon in it, it is organic, right? Because carbon containing means organic. And then that carbon there is single bonded with two other oxygens, okay? And then there's a hydrogen that comes off of this oxygen. And you may have learned from physical science about ions or charged molecules. And so that's what that is. So that's what it looks like. So this is bicarbonate. This is actually what allows us in our body, in our blood, not to like totally turn acidic or totally turn basic. It actually maintains a relatively stable pH because of this bicarbonate system, okay? All right. So how does this apply to our lab? Well, in our lab, what we're going to do is we're going to have a chemical called HCl, which is called hydrochloric acid, okay? And then we also are going to have another chemical called NaOH, which is sodium hydroxide, okay? So this one, the HCl, is the acid, okay? And then NaOH, or sodium hydroxide, is the base, okay? And you're going to actually add so many drops, no, you're not doing that because we had to do it COVID style. So there's going to be substances at normal, and then a beaker of that substance with 15 drops of each one of these. So one will be sodium hydroxide, 15 drops. Another one will be hydrochloric acid, 15 drops. And then there'll be two more beakers with 30 drops of each. So there'll be a 30 drop NaOH and a 30 drop HCl. And what you're going to do is you're going to use these little papers, and you're going to actually take some of the substance and put it on the paper, and the paper changes color, and whatever color it changes to indicates to you what pH it's at. So you guys are going to be collecting data on different substances and the impact of an acid on that substance and an impact of a base on that substance. So you're going to collect that data, and then you're going to analyze that data and see which ones or one is more like a living thing and how they would react. So what we should see is like the living things, or what once was a living thing, not react as much, right? Because living things produce natural buffers, and so they have the ability to kind of reduce the impact. So hopefully we won't see much change in those things, okay? All right, so this video is for you to watch either before the lab and or after the lab, or all the time, okay? All right, and that's it.